Now, this really today is going to be our what I would almost call the news update, the news and views update for UK NEQAS Haematology. We would normally give this usually after lunch at participants meeting, so you'd all be sitting there trying to stay awake after eating your sandwiches and drinking your coffee or what have you. I'm hoping that you have all had lunch and or whatever, because you may be from different parts of the world. But this really is an update on what we're doing within the scheme and within the programs that support the scheme. So I would give a brief overview of our automated haematology programs, our hemoglobinopathy programs, and our morphology programs. Please do ask any questions. I may not be able to answer them all at the time, just simply because sometimes they do require a certain amount of data or a certain amount of examination. But anything you do ask or any suggestions that you have will be taken on board, and we very much value the feedback from participants to these webinars, as we do to our annual participants meetings. If anybody can remember when we used to be able to do those kind of things. So in terms of participation, and this is data that we're very familiar with within UK NEQAS, but we are aware that participants don't see this all the time. We currently, within the scheme, have around about 1,800 laboratories that participate with us overall. Since 2012, which is 8 years ago, this has now gone up by about 50%, as you can see, from about just under 1,300 in 2012 to 1,800 today. So this is a considerable increase in work, which is good for the scheme, and we're pleased that it's actually it values the profile that we provide. When we look at the actual numbers of participations, that is even higher. If we look at the number of individual participations rather than laboratory participations, this is the total number of participations in all the various schemes, we have around about 6,500 or just over 6,500, which is a lot of data and a lot of accounts and a lot of details to try and manage. So I hope that we actually give you the service that you need, but I hope you will realise the amount of participations and the amount of performance that we are actually trying to manage. And on top of this participation in what you could call our wet schemes or our practical schemes, we also have over 4,000 individual practitioners in our digital morphology programme. You can see over the years, and this is over about the last 10 years, that there has been a change in the profile of our laboratories. If we go back to 2009, 2010, just over, around about a third of our participants were UK National Health Service laboratories. That is now just over 20%. That reflects two things. One is the consolidation of pathology services within the UK, but also the overall increase, and the large numbers of increases come from our non-UK laboratories, our non-UK colleagues, which is now forms just under 50% of the total number of participations. We also see an increase in the number of point of care testing, even within the NHS, and we separated these out because I think it is a useful number to observe. And you can see now that we're almost at 20% of our participation is from NHS point of care testing. Many of these private laboratories will also be POCT schemes. So I think it's useful for us to understand, and for all of our participants to understand, the profile of the laboratories that take part with us. If I then go to our automated counting programs, we have the bulk of all of our participations are in these programs, and we have, again, the largest ones are obviously within the full blood count, the automated differential counting, the retics and the ESRs. We saw a slight reduction in the number of participations for full blood count in the last year, and that did reflect COVID-19 pandemic, because we had a number of what I would call private POCT providers, in particular places like gyms and wellbeing centres that were closed as a result of the pandemic, and they withdrew from the programme or suspended from the programme during that time, but they have now bounced back again. But I think you can see there is a fairly healthy profile of participation there. Within the full blood count, we have around 2,500 instruments in total registered with us at the moment. The big users, the Sysmex laboratories have certainly the lion's share of the participations, with well over 1,000 laboratories using Sysmex instruments, or well over 1,000 Sysmex instruments in total. Following on from that, we generally have our the Siemens Avia and the Beckman Coulter and the Abbott and then the Horiba users. So again, a substantial number of laboratories, or number of instruments, this is individual instruments, a substantial number of instruments to manage. About 1,500 of those, so this is about sort of I don't know, that's 60% or so are UK, and the rest are our non-UK colleagues. Given that number, it's surprising how good the performance actually is. This is the number of letters that have been sent out to UK laboratories, and there's one for each year here since 2015, so over the last 5 years. You will see this graph here shows the number of non-returns. We send out around about just over 100. These are first letters for laboratories who have failed to return for 2 out of 3 consecutive distributions. So which out of the number that are registered is really not a large percentage. And just bear in mind that this is over an entire year. The largest proportion of this is sometimes the POCT sites, which do struggle to return data to us in the same time frame that the large laboratories do. And I think I can understand that. If you have nurse-led clinics or clinics being provided in say mental health units, you can understand that the staff there sometimes have other things on their mind than returning their EQA. In terms of the analytical performance, so this is people that are actually got out of consensus performance, we've sent somewhere around about 30 or so letters over the last 5 years. Is 35 letters. First letters have gone out for unacceptable or out of consensus analytical performance in the full blood count. You can see that far fewer numbers, or there are far fewer, there's only about 10 to 15 that will go out that are then followed up as persistent unsatisfactory performers. So again, overall, I would say that the performance is quite good. This will, of course, also cover all those participants that have dealt with problems with sample handling, transposing samples, not mixing samples correctly, etc. Within the automated counting, the activities that we've undertaken in the last year have been fairly many and diverse, as well as running our routine services. Participants in the ADLC programme will note that we have started to roll out nucleated red blood cell counting as a parameter within ADLC. And we hope within another webinar, and in particular for some user group meetings, to follow up on the performance there in more detail. We've extended the closing time for the hemoglobin only programme to allow point of care testing sites more time to return. We've run, or we're in the process of running, pilot exercises for the HemoQ instruments for the WBC and Diff programme, because this has been a request from both participants and from the manufacturers. We've looked at ESRs. I'm not actually going to talk about whether we ask people to put citrate or not to put citrate in to the ESR samples, but we have undertaken a study there, which we're currently analysing. We're also looking at whole blood materials for Elifax ESR technology, and this is likely to come on board more quickly. We've undertaken a pilot exercise to look at the provision of EQA for retic hemoglobin. This will be covered more in the third webinar in this series by our as chair of our steering committee, Dr. Wayne Thomas, who is going to be talking about the application of some of the novel parameters in full blood counting. The infectious mononucleosis program is it's still a pilot at the moment, but we are just about to submit it for addition to our UCAS scope of accreditation. And I wanted to talk a little bit more today about how we're scoring that. We're also, and Yvonne in particular has been looking at a review of handling instructions with both participants and manufacturers, particularly in relation to users for the UMIZEM 500 analysers, for which there's a little bit of confusion at the moment over how they should be running instruments, so how they should be running EQA specimens through their instruments. And I would ask them to follow our instructions fairly to the letter, because it's the only way we can actually gather sufficient data to make an evidence-based decision over how specimens should be handled. So what I was going to talk a little bit more about today, and I would just for each of our groups of programs just take a couple of items to talk about in slightly more detail. And these particularly are the use of the whole blood materials for the Elifax ESR technology and scoring the infectious mononucleosis screening program. Just one slide on the Elifax ESR. Those of you who have Elifax ESR technology will know that we're currently distributing latex suspension as your EQA. This is not ideal because it's not what you're testing for patient sample, but it is just really and truly some slightly different versions of the calibration material. However, because of the nature of the material, it gives a very good CV. The performance is excellent. We are, however, trialling the use of whole blood commercial material, which is provided by a subsidiary of the R&D Systems company. R&D Systems is where we obtain most of the commercial material that we use for programs such as the automated differential counting. We've provided one exercise to date, and we have compared that with some results from an EQA provider that uses the same material in France. We did get comparable results, although they weren't quite what we expected from the certificate of analysis. But interestingly, our colleagues in France obtained very similar results, which was reassuring for us in any case. Because of the outcomes, we have decided to distribute a second parallel exercise this autumn, and that will be going out. Shortly alongside the latex material that you're currently using. So please do return both of those results. Although this material is more ideal for EQA because it is a whole blood-based material, the CVs will not be as good, I'm afraid, as the latex material, but I think they will be a more realistic representation of the performance of the technology and the technique, and more in line with what we see for the other ESR instruments. But I think participants must steel themselves in a way to suddenly see the CVs going from around about 5 to 10% up to more like 20 to 30%. So, you know, just please be kind to us over that. The other thing that if we do go to using this material that we will have to look at is the overall program design. In the use of a commercial material, we are very much tied to what the manufacturers of this material can provide for us. This material is manufactured 3 times a year, so we will be changing the frequency for the Elifax ESR instruments, not for the others, but there will be separate modules, and they will be running on a different frequency throughout the year. That is going to provide a bit of a nightmare, a bit of a headache for us. The participants will not see any change to their reporting or the layouts or anything like that, but it will mean that there will be a slightly different scheme design. Going on from that, the other thing that we've been working quite hard on this year is getting the infectious mononucleosis screening test up to be as fit for UCAS accreditation. It is just about there. Really and truly, how long it takes to become accredited will depend a little bit on the workload for UCAS. Once we've submitted it, it is very much, and it's been inspected, it is very much at the mercy of the workload of the assessors and those that review the assessment. However, you will be aware that we've been sending out material. It's generally got good feedback. We see very few problems. The last piece of the jigsaw really has been to look at the extending or developing scoring. You will note that there has been scoring in your reports. The scoring at the moment remains on a shadow basis. It would not be reported or escalated to any of the oversight bodies. One of the things that we have puzzled over a little bit is the use of how to score what we would call, what one would term weak positive samples, you know, those sera that have a lower titer. And if I look particularly at it was 2003 MN, where there was a specimen in that particular distribution that was a weak positive, and as a result, in that distribution, we had 30 out of consensus results, because these are obviously borderline samples, compared to about 10 to 15 for the usual mixture, or in this particular survey here, which was 2102, where we sent out 3 negative specimens, or if we sent out 3 strong positive specimens, everybody got it right. There were no out of consensus ones at all. And of course, this does affect the cumulative scoring for participants. And you will see here that there were 30 here, and the percentage of laboratories that were actually had a cumulative score of 50 still actually went up for the following one, because the score will roll forward. Overall, in terms of persistent unsatisfactory performance, the performance is very good. There is only kind of 1 or 2 in any particular at the end of any particular distribution that has still had a score of 100, which would indicate persistent unsatisfactory performance, and nobody has fallen into the category of needing escalation any higher. We discussed how to score the borderline samples with our steering committee, and there were really 3 options. We would reduce or very much restrict the number of weak positive specimens that were distributed, only to maybe put out 2 annually and not in sequential distributions. This would smooth the impact of the weak specimens over the year and would not make a laboratory a persistent unsatisfactory performer for having a couple of weak specimens in a row. The steering committee was really against this, because they felt that it was very important to measure performance with the samples that are more borderline. The second option was to increase the consensus requirement for a specimen to be included in cumulative scoring up to 90%, as opposed to our usual 85%. Thus, we would still be able to retain the impact of the educational impact of sending out the weak positive specimens, but we would reduce the impact on scoring. This really is we were somewhat unhappy about, because it does mean that we would have to be withdrawing more specimens than we would like from scoring each year. The final option was to reduce the adverse penalty score to 30 or 35 for a weak positive specimen, to reduce the impact on the cumulative analytical performance score. And this was the choice of the steering committee. They also suggested that we considered adding a clinical assessment step and the next steps question. So looking at this would change the scoring slightly, because then there's the question of how do we decide whether something's a weak positive specimen or not. So we decided that if there was a greater than 90% consensus that the specimen was positive, then that would be considered not a borderline specimen. And if you got it wrong, basically you will get a 50 penalty adverse penalty score. If, however, the consensus was between about 85 to 90, which would suggest that it is a more of a borderline specimen, that score would be reduced to 35. And what that would mean is that you would need actually 3 specimens in a run of 3 distributions for you to become a persistent unsatisfactory performer and get a score of greater than 100, or 2 specimens to push beyond the unsatisfactory performance limit of 50. If you have a negative, if the consensus were negative, well, if it's more than 85%, then, and you called it positive, well, that would be out of consensus. And if any specimen had a less than 85% consensus amongst the returns, it will be withdrawn from scoring. And this is a fairly standard cutoff that we use. If we send out a specimen and there's less, and it is a qualitative specimen and there's less than 85% consensus, we do usually withdraw that from scoring, unless there is something like a PCR in the parasites, where there is a gold standard correct answer. Because, you know, clearly if we send something out that's got less than 85% consensus, then maybe the sample is too challenging, or something has happened and there's been some deterioration. And I will say here that 85% cutoff is for the individual specimen, not for the whole survey. So that is what we will be going forward with. Obviously, we will review this after 6 months to a year, but that is what is going forward and what you will see reflected in your reports. So what else is still to come with the automated counting? Well, there is an outstanding request. This is a participant suggestion for performance for improvement, that we include percentage bias in reports, which is on our IT list of tasks to be done. We have been looking at optical platelet count data that we've been collecting for Sysmex instruments, and a member of the team here has done some analysis on that, and we hope to report that back to people. We're looking very much at development of programs for the new point of care testing technologies. This is in addition to people like to instruments like the HemoQs, but some of you will be familiar with instruments such as the Olo machines from Cyte or Pixel, which are using an entirely different way of working. And we are working with the manufacturers and participants, and we have a task and finish group set up to see how we can provide EQA for these new technologies. We've had a request from the steering group to start to look at other parameters such as RDD, RDW and MPV, which we've also already done some work on. We've had a suggestion that we need to introduce interpretation and next steps in both the ESR and a next steps instruction in the plasma viscosity. The plasma viscosity already asks for an overall assessment of the sample, and we should be introducing that into ESR and asking what people might do next, which will provide us a little bit more of educational insight into the programme. And some participants will have noticed that we do have two alternative platforms now for data entry. There's our standard KPMD data entry. We are also using an in-house platform which is used currently for the infectious mononucleosis and also the blood component monitoring program. And our IT manager is developing a universal login, so you don't have to have two separate logins. You can have one login and you can then drop into whichever data entry platform that you require, which we're hoping will make the system slightly more user-friendly for participants. Please don't now all shout at me and say, why can't we do that for all our UK NEQASs? I can only run my own scheme, I'm afraid, but I do understand your frustrations with some of that. To then go on to the hemoglobinopathy programs. We have our abnormal hemoglobins program, which has 3 components. The newborn screening for dry blood spots, the DNA programs, and we also include in this our red cell enzymopathies. The biggest schemes are the abnormal hemoglobins, including the sickle screening only. And I think something that is significant now is the fact that the non-UK participants do now exceed the number of UK participants in that programme, and I think that reflects the profile of this particular programme, which is very highly valued overseas, but also the reduction in the number or consolidation of the number of centres undertaking abnormal haemoglobins work within the UK. In terms of performance matters, performance is really quite good. This is unsatisfactory performance letters spent out over the last 10 years for UK laboratories. You can see that probably in the last year, this is for every 6 months, for the last year, there are only around about, well, there's actually fewer than 10 first letters, hemoglobin A2 and hemoglobin S performance. The biggest number with about sort of 10 to 20 each year is for non-returns, and a certain number also for sickle cell testing. No, sorry, for actually non-returns. I'm misreading my colours here. Not for non-returns. Non-returns are very, very low in this programme, but actually for the sickle cell testing and fraction identification. And they often relate to sample transposition, is one of the biggest problems. Sample transposition and testing last month's or last distribution samples is the biggest reason for errors in this programme. We do maintain the all laboratory trimmed mean for both HbA2 and HbS percentage, and I am aware this has caused some concern for some participants. However, despite those concerns, we have actually sent out only about 5 to 10 first letters for hemoglobin A2 and hemoglobin S performance in 2020. We have, however, discussed the clinical significance for a cutoff for scoring hemoglobin S with our scientific advisory group, and they have agreed that we need to adjust the scoring so that we are really only picking up those people that are more than 3% away from the target, as that being a clinically acceptable variation, a clinically acceptable cutoff. This will require us to change the weighting factor, and you must forgive me, I haven't quite got to the amount of work that's needed to do that. We will need to reduce the weighting factor, which will reduce the impact of your deviation index on your score. In terms of overall activities, we did have some quality incidents around the, around the quality of a couple of batches of newborn dried blood spots cards that were sent out, and we have improved the minimum quality standards for those. We've actually been working with actually an American consortium to provide sickle screening to central screening laboratories in Africa, which is again a very high prestige thing for the scheme to be selected to do that, and we were very delighted to be doing that with international colleagues. We've been working on the production of cell line DNA for DNA hemoglobinopathies. This is a long-term activity, but it is going well. I've already talked about the hemoglobin S performance limits. We are also developing scoring for interpretation in the abnormal hemoglobins program, and we've been continuing to work on interpretive scenarios to support the aims of the NHS sickle and thalassemia screening program. And they're the two items I just will look at a little bit more. The NHS sickle and thalassemia screening program, for those of you that fill out or see family origin questionnaires, you will know that one of the first questions that is now asked when these are filled out is the pregnancy the result of egg donation? And this has become, in screening these new mums is quite an interesting or challenge, because obviously the mother's own hemoglobinopathy screen does not predict the risk to the baby that she is carrying, because the genetic makeup of the baby will be different. And it is important, therefore, that the baby's biological father is tested at the same time as the mother. We've sent out 3 scenarios now over the last couple of years that have examined this, and this basically is, what I've said correct here, these are laboratories who have either used the test the baby's biological father code, or in a written comment have said, you know, this lady is pregnant by egg donation, we need to test the baby's biological father. You can see out of all of the participants that only about 50% reflect that requirement. But if you think back to the number of non-UK laboratories, this is where the impact of the number of non-UK laboratories can be seen on the figures that that we report. And it's why within our reports we do try to point out where, or the number of errors that have been made by UK laboratories. Within the UK, where people are far more used to using the sickle and thalassemia laboratory handbook, you can see that we've got a far better compliance, and we are up around about, it was 80% for the first one. I think we wrote a little bit in the report, and people have obviously cottoned on to this a lot more now, and we were up to about 94 or 90%. This is never going to be 100%, because there would always be the ones that are missed here are laboratories who maybe couldn't return, or they may be laboratories that don't actually do antenatal screening. But it is good to see these figures, this compliance up to about 90%, and it's where it should be for compliance with the sickle and thalassemia program's direction. The It will also assist us to going on to the second item I just wanted to look at briefly, which is scoring interpretation in the abnormal hemoglobin surveys. You will all be aware that we've been asking for interpretation for a number of years now, and it is reported back just for educational purposes. We have, however, set up a task and finish group to actually look at how we can implement scoring, and it does need us to go one step back because we need to look at how we're capturing the data. So far, the task and finish group have reviewed the free text comments, which are also sent in. We've reviewed the comment codes. We've removed some, and people might be glad to see that we plan to remove things like no sickle hemoglobin present, no evidence of thalassemia, because these do cause some confusion, both for us when we're doing the analysis, and I think also for laboratories. And we've added some new codes to take account of the free text comments that people use. In order to score interpretation, we've decided the best way of dealing with this is that we will have a model answer that is provided by an expert panel against which laboratories will be marked. And we've designed a pro forma for that. We've recruited some test sites, half a dozen or so test sites, in order to help us test this program. We would be interested, if anybody is more interested, email us or pop something in the chat, or the Q&A, and we'll pick something up from you, because we'll be able to, if you just pop your email, well, perhaps not pop your email address in the Q&A, but just email us and we will be able to pick people up. What we will need to do, however, also, in order to make sure we're judging laboratories correctly, laboratories will need to be split into what we've called level 1 and level 2 laboratories. So level 1 will be laboratories that really only have one screening method available. So you are a first-line screening laboratory that has either just HPLC or capillary electrophoresis available. So you don't have it, so all of your samples that require confirmatory testing are sent away for testing. So what we will require you to do is to report your screen and then tell us what you would do. So it is a kind of a what do you see and what would you do. So we will have some more detailed options here about, the sample will be referred, or we're unable to differentiate these two variants, or there's an abnormal variant there, and it will be sent away for confirmation. And whether you would, for example, request testing of the baby's biological father. And of course, the whole idea here is to see that laboratories are operating safely and that UK laboratories are operating in compliance with the sickle and thalassemia programme's guidance. The second level laboratories are those that actually undertake more diagnostic testing and will also undertake confirmatory testing, so they have more than one screening method available. So these laboratories, again, it will be still a what do you see and what do you do workflow, but they will be expected to screen, do confirmatory testing depending on the primary screening results, and then to provide us a report. So, and these two will be, again, you'll be scored on, have you given the safe response for the patient? But again, we will not be scoring these laboratories against the same criteria as the level 2 laboratories. And I think that will make it much easier for us to identify and correctly score laboratories. So still to come, again, not really hemoglobinopathies, but they're the enzymopathies. Those of you who undertake the G6PD program will note that we have been sending out more human G6PD deficient blood over in recent distributions. That has partly been brought about of two reasons. One is we have identified a better panel of donors for G6PD carriers. The second fact is that with the changes to import and export following the UK leaving the European Union, actually sending a package with mixed human and animal bloods in it into some countries is a challenge. It's always been a challenge outside the European Union, in particular, if you're going to places like Australia or Peru or anywhere like that, because sometimes the approvals for these imports go through different ministries, and having them in the same package, it just becomes a logistical nightmare. So we've been trying to source a better panel for human G6PD deficient donors for that program in place of sheep blood, and we feel we have established that. And I think, I mean, obviously that is a much better move for everybody, because you're testing human blood, not most of us, I don't think, are in clinical laboratories that are testing sheep. So, you know, it is much better that you have human blood available. I will also say that the pyruvate kinase pilot, there's very few laboratories in the UK that do this, but I know internationally it has been popular. It will return. It was one of the things we had to hold back as a result of the pressures of the COVID-19 pandemic on our own team. We had to look very carefully at what we could sustain when we had staff that were either having to work from home, having to shield, or were unable to come to work because they were isolating. We had to make sure that we protected our core services. So going on to the morphology programs, as you would expect, our biggest programs is the blood films for morphology, for differential counting, and parasite identification. We still have a significant number that's doing the cytochemistry for the hemosiderins and the rapid diagnostic testing in malaria, which is not really anything to do with morphology, but it goes out in alliance with the blood parasites, is also got a significant number of participants. So a nice healthy number of participants there. The digital morphology program also is looking very, very healthy. We've got over 4,000 licenses across 340 laboratory accounts. And we have, in addition to that, we have another 300 individual users, and these are people that subscribe to us, I don't know, just as individuals, presumably because they can't get a license supplied through their workplace. And we now supply, this service is now picked up primarily in the UK, but in 26 countries in total across the world. So it is a very popular and obviously valued program. As those of you who take part in this program will know, we transferred platform a couple of years ago. This was a matter of some concern for us. Providing the new platform was a significant investment in both time and money, and you know, we were concerned at that point that the number of people would drop off, the number of participations would drop off, but in actual fact they've gone from around 3,000 up to about 4,000. We've also noticed that we see a higher proportion of returns for every individual case. This looks slightly lower here at the moment, but this is numbers to date. And we find at the end of the year, the average by the end of the year tends to be up at around the 2,000. And we hope this reflects the fact that people are finding the new platform more user-friendly or whatever. But anyway, we're very, very pleased to see the profile that's being maintained in the digital morphology. Overview of work that's been going on. Well, we've also transferred the hemosiderin EQA to the digital platform, just on one exercise. We have, as you will appreciate, introduced images into blood film reports, and we hope that you find that useful and it gives you more feedback to have just a snapshot of cells that you should have looked out or for educational purposes. We are working on getting graphical representation into the differential counts. Again, we are waiting a little bit on our software provider to support us with this. We've also been working with colleagues in UK NEQAS Parasitology on the supply of slides for blood parasites. This has been impacted upon by the COVID-19 pandemic. The main reason for that is that nobody's been going abroad, and nobody's been coming back with malaria and going to the hospital for tropical diseases where samples could be taken. So what we are looking at, we're having to look at using smaller batches, and what this may mean is some participants who are registered for the blood films for morphology, but opt not to return results for the blood parasites, we will probably be removing the blood parasite slides from your package. So even if you did find them useful just to have them there, or you might have just thrown them away, we won't be supplying those. We're looking to dropping those from the future. We have also been trialling differential counting for what we've called automated cell identification systems, which is perhaps a bit of a shorthand for cell-vision type technologies. We've used this term automated cell identification systems so that we don't look to be preferential over any one manufacturer. And we've also been looking at the development of the EQA platform to look at consultant level morphology skills cases. And when I say consultant, we're largely working in collaboration with our medical consultants, but of course this may also be used for SPRs or in training, and also higher level biomedical scientists and health and clinical scientists who actually report out cases. I just took a little bit in a couple more slides for the hemosiderin EQA and looking at the trialling the automated counting by cell-vision or automated cell identification systems. Those of you who take part in the hemosiderin cases will note that we have sent out one exercise now with digital images. This reflects the fact that we can no longer supply decent blood or decent bone marrow slides for staining for hemosiderin. You will all be aware of the requirement really for a good bone marrow slide for hemosiderin to have 7 particles on it. We felt we were lucky to get 1 particle on even occasional slides. And the other problem is actually obtaining any bone marrow, even hemodiluted bone marrow in the first place, because so much of it now goes for markers, cytogenetics, etc., that there is little left over. And of course, there are fewer bone marrows actually being done. So we are going over to using digital images, and we piloted one through the EQA platform. We are very much aware of the fact that the EQA platform, the actual access to the slide was less than ideal because, and I do know from lab managers, we have noted that you weren't sure who should be reporting it, and that everybody that was registered for EQA in your laboratory was actually receiving emails. We're working on that with Certus Technologies, who provide the software for the digital programs, and we hope that that will be fixed by the time we send the next distribution out. But this will be the way forward. We've also had feedback about the fact that it doesn't test the staining, and we are aware of that. The second phase of this development will be to look at sending out some form of slide, and this may be urinary hemosiderin, it may be some alternative tissue, that we can send to you to test the staining. And we're not sure quite how we will assess that. We may ask for slides to be returned to us so that we can then assess staining. But we are aware that it is still essential, and especially for UCAS accreditation, for us to supply you with some way in which you can actually test the staining. The other comment that has come out, which we think is in response to the digital slide, which we think is also important, is the provision of the image of a control slide to go with the hemosiderin stain, the cytochemical staining, and I think that needs to be taken. We will take that on board as well. So thank you for your feedback, thank you for your patience, and we can only develop this in conjunction with you as the participants. The other thing that we've been looking at is trialling the automated WBC differential, and we started this actually with Cell-vision themselves, sending slides out to Sweden to get them to look at our slides and whether we could just use the same slide through the Cell-vision as well as a manual microscopy. And largely it does work. It does depend a little bit on the software that you have with your instrumentation. But we have had one trial that has already gone out. We invited around about 30 labs, and I think we had about 20 that returned, and we are preparing a report on that. But it will allow participants to send in white cell differentials, both using their manual microscopy and using their automated cell identification system. And we will probably trial the automated systems both before and after any manipulations or any reassessments that have been done by the participants, and report those back. And of course, if you happen to get a slide that won't go through in the automated fashion, well, we won't penalise you on that. You'll just have to revert to manual, I'm afraid, and use a microscope. So, you know, let's hope that that will go forward. The Bloodfilms Morphology scoring will come. We've been promising this for several years. Again, we are very dependent upon our software provider, who again, I think have been held up to some degree by the COVID-19 pandemic, but it will come. We have agreed all the criteria. It's all been agreed through our scientific advisory group, and it is now just waiting for the software to be written and the verification, validation of that software. So, you know, hopefully you will start to get feedback on your Bloodfilms performance. So just going on just finally to the last couple of slides for news and views. One sad thing I will raise with those of you who knew Professor Machin. Professor Machin died a couple of weeks ago after a short illness. Those of you who have been longer hands with UK NEQAS will remember that Professor Machin was the chair of our steering committee from 2006 to 2009 and was a great supporter of laboratory haematology and laboratory in general and haemostasis, and a great supporter to more junior colleagues and promoting research in laboratory haematology. So we send our condolences to his widow and his family and friends. Oversight of EQA performance. The Royal College of Pathologists is leading an initiative to harmonise and standardise EQA oversight by setting up an EQA oversight board and stakeholders committee and some work streams looking at governance of EQA oversight and governance performance standards. The intention is to standardise not so much what you might see as participants, but the operation of the NQAPs, improvement of communications between those bodies to whom performance is escalated, and a better means of looking at performance across different UK NEQASs, across different EQA providers, and especially when we're looking at problems that may have cropped up that are method related. And just a little change of terminology, you may notice that the Joint Working Group on Quality Assurance has now been replaced by a different committee, but similar makeup, known as the Quality Assurance in Pathology Committee. Final little bits, we are moving amongst all the other things we're doing in a month's time. We are moving from the 22nd of November, and we're only going to go, we're going about 1.5 miles down the road to a business park near the hospital. Unless you come and actually visit us, and people are welcome, you won't notice a great deal of difference. The PO box address will remain the same. We will have new phone numbers. We will remain part of the West Hertfordshire Hospitals NHS Trust, but this move, this relocation to commercial premises is a requisite because the Watford General Hospital site is undergoing a massive redevelopment over the next 5 years, which means that the whole site will be being rebuilt and redeveloped, and as part of that, any what almost called back office or non-core functions are having to relocate off-site. And finally, last but not least, we do intend next year to get to Liverpool for a proper participants meeting. We tried to get there in 2020, but that was obviously stopped by the COVID-19 pandemic. We were trying to get there this year, but we still felt that people will still be cautious about travelling. Also, the idea of actually running a whole participants meeting at the same time as relocating the entire department was really a little bit too much to bear, and we've resorted back to 3 series of Zoom webinars this year. So there were 3 earlier in the year and 3 now. So at least 6 hours of educational feedback. But we do hope next year that we will be able to return to a face-to-face meeting. Now, what would be useful to us, I think, as meeting and educational service providers, is to know from participants what you would like in terms of participants' meetings. You know, do you like the options of webinars? Would you like blended? Content? Are you all delighted at the notion of being able to get out of the laboratory for a day and go and meet up and network with colleagues? Because I think the way of providing these kind of participants meetings has changed and will change. And if anything good can come out of the COVID-19 pandemic, we need to look at some of these new ways of being able to interact with our participants. So do let us know. And that is basically it. So thank you for your attention. And do we have any questions? I need to go look at the Q&A.
Thank you, Barbara. And I'm sure, oh, I forgot my mic up. I always do this. Thank you, Barbara. I'm sure everyone will agree that that was a really good presentation. And yes, we do have some questions. So I'm going to start with a question from Austin, which has been voted by a few people as well. Austin has asked, are there any plans afoot to update the online results portal? For example, if the system could email you maybe the day before an EQA submission date, as a reminder would be excellent. Also, if the system emailed you when reports for an exercise become available, similar to the EQA platform. I'm sure we've all in the past had a mad rush at 4 p.m. on submission day trying to get something in because it slipped off our radar.
Yes, Austin, I'm very much aware of this. I think actually it's a really good suggestion. I mean, in terms of reports, the contacts you are emailed when the reports become available, but I do take on board the notion of knowing when the submission is due for particular programs, especially since the submission dates are now varied. Once upon a time, they're all the same day, and you either got them in. And I do know that mad rush. At least nowadays, you aren't running around trying to look for a working fax machine and trying to plead with your colleagues in blood transfusion to allow you to use the blood. What is the blood? Only the blood order fax. In order to actually get a working one. But I won't say that we have any plans afoot right at the moment. I can't promise that, but I do think it's a good. I think we will take that as a way, as a suggestion. You will know obviously from our website that there is, you can get your own personalised calendar now, but obviously that does require you to go and look at the website. And it may be that we can do something that's outside the actual submissions portal, but I will speak to our IT people who run the website and who set up things like the personalised calendar for the report for when surveys are opening and closing, whether there is something that could be done there. Again, I can't promise, but I do think it's a very good suggestion, and I will add it to our participants for suggestions for improvement.
Thank you, Barbara, and thank you, Austin, for that question. And we have a question from Curtis. Would you be able to comment on UCAS's view on the commutability of the latex material for the ESR EQA samples?
I think in a very short answer, no, because I don't think I've ever actually asked UCAS. I mean, the samples, are the samples commutable? They're clearly only suitable for Elifax machines. So they're therefore not commutable across any other platform. Whether UCAS feel they are suitable, I don't know. And I don't know whether you've had any feedback from UCAS or any queries. If any participants have, then I'd be really, really interested. If they can drop me UCAS's views or drop our general email web address, any views that they've had back, it would be very useful. But I've not discussed this with UCAS, and UCAS they certainly haven't criticised our distribution of latex material. And of course, the kits that we provide are provided for EQA, and they are provided as EQA rather than just the same materials that are going through as calibrators, although it is the same formulation. But I do know that they are not particularly, well, the participants feel that there should be something better. So we're looking at whether we are able to deal with that. But thank you. And if you've got any feedback, do drop me a line. I'd be very interested on that.
Thank you, Barbara, and thank you, Curtis, for the question. I have a comment from Romania. She said, we would really like a hybrid webinars in the future to allow non-UK participants to participate. So thank you. Oh, and she's popped in another suggestion. Can you kindly run webinar series for non-UK FRC path candidates as well?
They're excellent suggestions, Romana. I'm not sure whereabouts in the world you are, but I do feel that these hybrid webinars in some way would be a good way forward, because it does allow us to reach more participants. And what we're also looking at as an option is regions or countries where we have a distribution agent and we have a substantial number of participants. We are hoping that we might be able to put, oh, you're from Pakistan, you are welcome. What we are looking at is whether we could provide webinars for a particular country, so especially where we have a particular distribution agent. So we may be able to link up with participants through distribution agents. I'll take on board the notion of non-UK FRC path candidates, and again I'll have to go to colleagues at the RC path and see whether there is something that could be run, or maybe again you could drop us a line and see what you would like for certainly the EQA provision for the RC path. We'd be more than happy to look at that.